For our next presentation, the presenter is Dr. Mantos Pano. He is also related to the SART institution, specifically KeyLab, and he is recently graduated from his PhD, and he will talk a little bit for us. Mr. Dr. Mantos Pano, can you can go? The screen is yours.
Okay. Um, can you see my screen?
Yes, we can see it. I think it's not in the first page.
Yeah, me too. Okay. So, hello everyone. Today I'm gonna present you instead of Mr. Svetsas who cannot attend here. So our presentation will be focused on a biogas lab operation and the main characteristics and the utilization of digestate. As you probably know, in the biogas sector, we have different inputs as a feedstock, like energy crops, plant bioproducts, animal products. And through the process of anaerobic digestion, the biogas plants utilize either the digestate as a farming product, or they can produce a different kind of products like biofuel, biomethane, electricity or heat that can be used for different purposes in our society. So, okay. So here's a graph of the number of different biogas plants in Europe through the years. As you can see here in the graph, there's a growing number of biogas plants established in Europe. And also in the map, you can see where they're in Europe. In Greece, we have like five biogas plants that are currently working in high numbers. We'll talk about it later. However, we also have a growing number of biomethane plants in Europe that are working again with the biogas plants, which is also due to the use of the potential use of biomethane. So currently in Greece, you can see here the registered members of biogas plants in the Hellenic Association of Biogas Plants Producers. I will show you an example. I will explain you later how is the process from the plant to the lab. Here is the example of Biogas Lagada, which is established in northern Greece near Thessaloniki. It's been one of the highest, the biggest biogas plants in Greece, and it's currently running in high power. So usually a normal day includes the transport of liquid cow manure, for example, from farms that are nearby or other companies by the company-owned trucks. They wait every day the loading, and they store it in liquid substrates and underground pre-tanks. They had also solid substrates, mostly from dry cow manure, and also from different kind of inputs like corn silage or other crop silage stored in this kind of pits that you can see here in this picture. The whole digest can be utilized as an organic fertilizer, and it's stored in two open storage tanks with capacity of 8,000 cubic meters. And there is also the possibility to separate the digestate into solid and liquid part. It depends on what kind of use the plant want to process with it. They have also a separate liquid digestate, which is pumped out of the underground discharge tank. in order to separate the liquid in the different forms. And they have the final measurement, and they have a gas combustion engine for to produce the biogas. So it's coupled with a generator for the energy production, and they produce low voltage electricity, which can be transferred to middle voltage electricity and can be used for the electricity that could cover cities nearby, like Thessaloniki, which is the second biggest, the second um, biggest city in Greece. I'm currently working in QLab. QLab is very closely related with these biogas plants because we running analysis every day for their digestate. QLab established on 2000 and, oh, sorry, 2012. And through the years, they managed to create new protocols, keep new machinery, and they have now very established methods for the analysis of different kind of digestate. Here is an example of the machinery we currently have. We can run analysis for both molecular stuff in that's going on in the biogas plants or the chemical analysis, and we can run some small scale experiments in order to see the biogas potential and the process of the anaerobic digestion. Yeah, here are some photos too from the machinery I explained earlier. What about the substrates? We're currently working with the characterization of multiple substrates, and here are some methods that we're using. For example, here we have two biogas plants that have different type of bio-wastes. In both the experiments, we have animal manure, and especially in the biogas plant one, we have cow manure. The first thing that we are doing is a chemical biomass analysis, and according to two different methods that we try to measure different parameters like pH, dry matter, lipids, proteins, you can name it, in order to have the best state analysis of the digestate that is coming out of the biogas plants in order to use for farming purposes. And apart from this, we run our own experiments in order to see what's the biogas potential in small scale, of course. We have BMP tests that we determine the biomethane potential and the anaerobic biodegradability. We run it every day, both from BAP test, and we have also two CSTR that we are using digestate that we taking from the biogas plants, and we want to see both the biomethane potential and the interactions between the different microorganisms that are taking place inside the anaerobic digestion. We currently calculating specific methanogenic activity during the BMP. Here are some graphs, some experiments from how we're calculating and the results we're getting. And for example, here we have the olive meal waste, which has an aerobic digestion duration of 40 days. So we're taking the inoculum and we try to measure all the parameters. And then, as you can see here in the different graphs, we are calculating the biomethane potential. Yeah, 75% of the total biogas yield was achieved after the end of the 8th day with a specific combination of microorganisms that we are using. Sorry?
Yes. Yes. Thank you.
Can I continue? Yeah. and the 95 potential of the biogas was produced after 20 days of the anaerobic digestion. We're also using other inputs like corn silage. The anaerobic digestion is the same, but we achieved here the maximum biogas potential earlier than the olive meal waste. So we also we want to characterize which input is better in order to have maximum result that we can recommend to the biogas plants. And yeah, we are currently working with biogas plants in horizon-based projects like Metaverse, where we try to explore the possibilities of the biogas, of how can do it for the gas processing and the storage, at the same time the storage of liquid digested and the solid digested. So we exploring new methods, new analysis, and new transformation on the final step of of a gas plant operation. Yeah, so here is the number of different research projects that we're currently working with these type of plants in Greece. Yeah, there are many of them, so I won't get in details for many of them. I have some one or two. So this is an example that we are working with the biogas plant Lagada, where we try to enhance, if you want, the biogas production by using different types of microorganisms through the anaerobic digestion. So we had the idea of using different supplements through the anaerobic digestion. So an example of that was the Evogen, which is a commercially product and has different consortia of bacillus. So we use it in both big scale and large scale in order to see what's going on and if we can have better results for the biogas plants. So yeah, the results were very promising, and it seems that you could minimize the cost at the same time, and you can have a larger biogas production and diesel and lesser diesel consumption. Yep, and this is the examples in the results in the small scale using the Evogen additive. So yeah, you can see here also in these graphs that if we add 1.5 gram of bacillus through the Evogen supplement, we have at the same time higher biogas production through every day, and I don't have a graph here, but also the input, the feedstock that we're using were less because this was our goal, all right, to minimize the cost. And another one of this project is the 3D biofilm, where we use 3D printers in order to produce shapes of different forms, different states that could be used in the biogas plants, and the microorganisms could utilize it and produce different consortia on it, and they can actually explore their capabilities and like for example, through the literature, we checked and we saw that some types of 3D carriers could utilize more methanobacteria on it. So we wanted to explore these type of things. And this is last the most updated, I think, research program that we are currently running right now. So it's called Me2Gas. It's actually microbe for gas, where we are currently taking samples of the digestate and the feedstock from the different biogas plants in Greece. We are running next generation sequencing in order to have a full profile of the microbiome. And at the same time, we're doing it in a small scale. We are trying to adjust some parameters like pH, temperature, so we can see which are the interactions and different relationships between different individuals of the microbiome. So we can have a hypothesis, or we can go to a Biogas plant and tell that you can adjust that, and you can have more methanobacteria growing that you currently need for your production, right? So yeah, it's a bit troublesome because many of the bacteria that we're currently trying to categorize are not classified through the databases. So yeah, we want to now explore more on it, like through some RNA analysis to check what's going on in the transcriptome. But yeah, for now we here are, for example, different type of bacteria that are making different processes through the anaerobic digestion. So we want to have both the profile, the number, and what's going on inside there if we adjust some parameters. Yeah, this is also what's going on inside the anaerobic digestion. So yeah, we are currently see that when, for example, the class Raminonatse is the one that it's more inside the highest numbers inside the anaerobic digestion, there's more energy produced. So we want to check how we can potentially amplify this type of organisms and enhance them in order to grow more in numbers inside the anaerobic digestion. So yeah, that's all. I hope I could present our results and our stuff in a good way because it was a late call. So yeah, thank you very much.